Thank you, everyone. Thank you for coming back after a lovely lunch. So it's an honor to be here and have the chance to tell you a little bit about our work related to psilocybin. I will be primarily focusing on the work done by two very talented individuals in the lab, Alejandra Pacheco, who just went for a very fancy editorial position, and David Jacobs. Some of the work, let me see if I can figure this out. There we go, okay. Some of the work that I will tell you is published, the majority is not. So let's get to it. I am not interested in, at least in this talk, in how psychedelics produce a trip or interesting stuff in animals. I've always been interested in, there we go, there's a delay here. This is not following, so pardon the slowness. The clinical effects of psychedelics, essentially. And what do I mean by that? Well, I've been interested in ketamine, antipsychotic drugs, and antidepressants, and how they work, and the lab has had many years of working in animal models related to these. With psilocybin, we know that some new papers have come out showing that they could have clinical efficacy. The best one was very much the fundamental work done by the group in Hopkins in terminal cancer patients, which very much put psilocybin treatment psilocybin use for treating anxiety and depression in the realm of legitimate, serious science. A bunch of other papers have been followed so far, including this high-profile one in JAMA a few years ago, actually showing that with a semi-placebo, psilocybin could actually be working to reduce depression. So a fair amount of studies are coming out, but there's also concern that psilocybin does produce side effects. It produces lasting fear and anxiety, perceptual disturbances. It also there's a huge concern, at least in North America, but I bet it's also the same with Europe. I haven't looked at the data. But because the recreational use is going up, there's also concern that the adverse effects and parapsychotic effects of these drugs could also be a factor. So with this in mind, we are, of course, interested in the clinical effects. The problem is, unlike years of studying a bunch of stuff that I have studied in the lab, such as antipsychotics, antidepressants, ketamine, etc., at least and I promise that most of you, have no idea if what works is the pill, the process, or pill and the process. Now, what do I mean by that? And actually, Alejandra wrote a small review on this. Essentially, with other drugs, such as, let's say, classical antidepressants, what you do is you give the pill to animals, an antidepressant to animals. You actually discuss, this is really not working, okay. You then look at the effect of cellular, subcellular plasticity effects, and then you try and correlate that with behavior. And then you deduce that, okay, that's why you're having X and Y clinical effects. In the case of psychedelics, it's not that simple. Literally all the studies that have shown a clinical effect have involved some level of clinician-assisted interference or manipulation, for the lack of a better term. So what is really important is that in this case, perceptual state and context and memory play a big role, and somehow then they interact with the drug, and at that point, they produce symptoms. Okay. So this is critical, but the question is then, if you are interested in understanding perceptual states, context, and memory in the context of pharmacology, how do we go about studying this? Where to start? And this is a question that has occupied my lab for the last 4 or 5 years, and I have to warn you that I will leave this talk with far more questions than answers, so be forewarned. Okay, so the question that we asked first was, what is the effect of psilocybin on active behavior when context, emotional valence, or cognitive load changes? So then with that in mind, we focused on goal-directed actions, so that the animal is actually engaged. It's engaged in getting a reward or avoiding a punishment, as opposed to spontaneous behaviors. So not head twitching, not like moving spontaneously in the maze. We want animals that are actually actively engaged in the task. And then, very importantly, focus on tasks that have very good, solid human analogs. Okay, so I'm going to actually focus on one that's published and then move to Kamin after that. So I have to emphasize that we use outbred rats. They are great creatures, but a rat is not a person. We are very much aware of that. However, rats are quite, I would say they're actually quite brilliant. They continue to outsmart a lot of us when we try to train them. They're not that anxious, which is very good. They work for at least an hour or more in operant boxes. If they're happy, they like what they're doing, which is very good for electrophysiology, because I'm not going to show you any electrophysiology data, but that we do a lot of that, and that allows us with a lot of trials. And nearly all the classical pharmacology data that we have with psilocybin, LSD that was collected in the 80s and 90s was done in rats. Okay. So with that in mind, one of the first things we were interested in is cognitive flexibility. It's almost a no-brainer why this would be interesting to the clinical field. Most prominent theories, I'm just giving this as an example, bring in cognitive flexibility into why psilocybin and these class of compounds work. For example, psilocybin has been theorized to reduce hyperpriors or maladaptive priors. Flexible thinking, again, has been implicated in why psychedelic work. And there's actual solid data, again, from the Hopkins group, that psilocybin treatment produces lasting improvement in cognitive flexibility. And last and not least, I am a hardcore neuroscientist, a systems neuroscientist. And for me, I need to always be thinking about actual neuronal mechanisms, systems, and not psychological constructs. And for me, if you are thinking about priors weakening or strengthening, that has to involve neuronal pathways that are regulating behavioral flexibility. So for me, that was almost a no-given that neural base of behavioral flexibility is key here. So the first study we did was it was actually a pretty thorough study, and we showed that acute psilocybin enhances cognitive flexibility in rats. I'm going to only show you a few data sets here. This is a pretty comprehensive pharmacological study, dose response with psilocybin, DOI, and a large number of other drugs. But what was really critical about this is that the task was very much a version of Wisconsin card sort. I'll tell you about it a little bit, in that it wasn't just simple shifting from A to B. It was a well-trained group of animals that learned to do multiple extra-dimensional shifts in the 30 minutes or hour that they were in the box. And it's then that we saw a clear effect with psilocybin. So the way the task works is, and this is published, we have published several studies with it. We have extensive recording from prefrontal cortical regions. So animals are in an operant box. They have two choices. One is correct, one is incorrect, and there are two rules. There's a light rule and there's a side rule. So the investigator depends which rule is correct. So if the light is correct, if they nose poke here, they get a reward. If they nose poke here, they don't. And then after they have 10 consecutive correct responses, we change the rule. And this can go on. We do a minimum of 4 shifts. Okay. So the way we did these particular studies, because the beauty of this task is that each animal is different, but they stabilize at their own basis. So we can do within and between subject design for the study of drugs. So the data that we primarily have here is once the animals have a stable background, stable session, we give them the drug, and then 20 minutes before we test them. And the gist of this particular study was that psilocybin actually improved shifting. Animals were able to shift quicker. And that's the data that I'm showing you here. So this is pre-psilocybin, pre-psilocybin, after psilocybin. So they take a fewer number of trials to reach criterion, and this is compared to saline. So this is well and good. After this study was published, we did a large amount of post-hoc analysis because we were trying to figure out why are these animals shifting better. And one of the interesting things we found was that they weren't like magically brilliant. There were plenty of errors in between. So psilocybin animals were still committing errors. They're just shifting faster. We also saw that the shifting was not at all dependent on whether it was the light or the side that was relevant, so that kind of eliminated the fact that maybe one is more salient, or whether it was preservative or regressive error. But the one thing that we saw was that we knew that response time was increased by psilocybin. The animals are just slower when they're in psilocybin, even though they were still complete the task. But when we look at their actual error and the correct responses, we found that successful rule shifting was preceded almost always by error plus a large response time. What does that mean? When we did the analysis, is that psilocybin produces post-error slowing. Now, for cognitive neuroscientists, this is a big deal because we've known, especially in the primate literature, for a long time that post-error slowing very much contributes with enhanced cognitive functioning, ability to shift. When you make a mistake, if you actually slow down and think about, what happened, that slowing, the longer slowing actually allows you to correct your mistake more efficiently. So post-error slowing, again, is attributed to prefrontal cortical systems. There's a large literature, and it has been attributed to the so-called retrospective contemplation or the so-called backward scanning. Now, these are loose terms for me as a hardcore neuroscientist. They're important psychological constructs, and the current memory field actually probably has fancier terms, but these are essentially the terms that were very much referred to before when we were considering how post-error slowing could be influencing behavior. So the question then came, does psilocybin influence this sort of backward scanning and retroactive processing? Okay, so this became the next question that we were asking a few years ago. So we decided thinking about how to test this. So just to quickly tell you what this involves, very simply, if you think about a cue that predicts an outcome, when you get that outcome, no matter how well trained you are and how predictable that outcome was, your brain is actually scanning back. If you're recording from prefrontal neurons, you still, you see a whole bunch of neurons that are getting activated to well-learned cues. And this sort of scanning back has to be there in order for you to keep track of priors, in order for you to be able to keep your priors and relax them, for prediction coding to happen, and for prediction error to happen. So this is essentially, this backward scanning is key for all these constructs that we attribute to potentially psilocybin acting on. And that, for good or not, brings us to Kamin blocking. Kamin blocking has been around since the 60s. I love this if you're into conditioning, you probably like it or not, but it has followings and haters and lovers. But it's essentially the learning that a stimulus predicts an outcome is blocked because a prior learning of that stimulus you have already associated something with that stimulus. So let me just give you some real-life situation here. And this is something that applies to me, so maybe not to you. Let's say you go and you're someplace in the lovely coast of France and you go and get fresh oysters, and you like oysters, and they're yummy, but you get sick. So you associate oysters at that place in making you sick, having salmonella. A few days later, you go back and maybe to another restaurant, but in the same area, and you have a salad and oysters, and you get sick. Okay. So your mind immediately attributes the oysters to being, causing you the illness. So a week or so later, if you go back and you have a salad, you're fine actually ordering a salad and eating a salad, even though your mind knows that fresh lettuce is actually one of the major sources of salmonella. Okay. So this is essentially Kamin blocking. that because you were exposed to this stimulus, to the stimulus before, that now your brain is focusing on this, ignoring that this could also be bad news. However, if you were exposed to both of them at the same time, this is a compound stimulus situation. So you're exposed to both stimuli at the same time, and you get sick, then the salad itself, I don't know, I have to decide which is which. Okay? So this is essentially Kamin blocking. So the way we run these in rats is that they're in an active operant box during lever pressing for food, and then we put them through several stages. As they're working, we expose them to a cue that is followed by shock. So when they actually learn that, when this cue comes on, they stop responding. And then once they learn that, we give them both the salad and the oysters, and then, of course, they freeze because they think, oh, QA was bad. Then after a couple of days, we only give them the salad, the QB, the second Q, and they don't freeze. And of course, obviously, you have a control group where they get both. In this case, when they get the salad, the QB, they freeze. And really quickly, I have just real-life data. This is our blocking group. The animals are responding here. They learn, this is the cue that predicts shock. They start responding. And then when they just get the second cue, the salad cue, they don't stop, whereas the compound stimulus does. Okay. So I'm going to, for the interest of time, I have more data if anybody wants to see it afterwards, but for the interest of time, I'm going to actually show you just a couple of psilocybin data pieces. So the data I'm going to show you will be a ratio. There's a ratio of responding when the stimulus is on, over, before, and after stimulus. What you should remember is that if this ratio goes down, behavioral suppression, so the fear of something bad may happen, goes up. So essentially, this is what the data looks like. This is, by the way, very much modeled after McNally's group in Australia, and this is how they run it, and works beautifully for us. So this is the animals. Suppression ratio goes down day 3 because they learned that the stimulus is bad. Here, the block group, they respond, but the other group that hasn't seen the stimulus, they get both the salad and the oysters here. They learn to respond. And then on the test day, 2 days later, they get the second stimulus. And the block group, they don't respond because they don't think that stimulus is associated with shock. Again, it's just like they don't think the salad is a bad idea. Now, this is a great task because it's very much related to cortical thalamic projection. So if you actually use DRETs, and this has also been done with lesion studies, to block this prefrontal cortical projections to the thalamus, then you interfere with the blocking. Keep in mind that the blocking was here. So if you actually inhibit the prefrontal thalamic projections, then the blocking goes away. So the nice thing about operant boxes that are quantifiable like this, we can also get to the circuitry. Okay, so what happens with psilocybin? The way we ran this was that on stage 2, when they got both stimuli, after they have learned that this stimuli is going to associate with shock, when they get both of them, they either get saline or psilocybin, and then 48 hours later, they're tested. So here, what we see is we always do dose response studies. I was trained as a rigid pharmacologist. So 1 milligram, 3 milligrams. So with the 3 milligram, this responding is different because the animals, the 3 milligram is pretty high. The animals just are not responding. However, what's important is that the day after we get the psilocybin, the behavioral is back to being normal. They are withholding the response. Okay. So then what happens with psilocybin is 48 hours, again, at this stage, and what happens is that psilocybin dose-dependently weakens Kamin blocking. So the animals are responding also, or inhibiting the response also to the stimulus, to the salad, that could be potentially predicting shock. And again, if we go back to this study, which we have inhibiting the thalamic projection, if we do the opposite, now we come and actually excite the projection from cortex to the thalamus. Now, these studies are preliminary, the N is low, so please, we're not ready for prime time with these, but I thought I'll show it to you. So if we actually do that, then we can reverse the effect of psilocybin. So if you actually look at here, this is our control, this is blocking, this is psilocybin inhibiting the blocking, but we can reverse that inhibition if we now activate prefrontal projections to the thalamus. Okay, so to summarize, pretty straightforward data in terms of what the data is showing. Psilocybin, while on board, while the animals are experiencing it, improves behavioral flexibility as measured by shifting action choices based on previously learned outcome associations. It doesn't do it to very simple cue outcome associations. If it's really, really simple, it doesn't do it. It has to be this kind of complex extra-dimensional shift for one reason. This improvement, we've done a lot of analysis to get to the bottom of it, but the only thing that pans out is that it's associated with post-error slowing of action selection. And also that psilocybin weakens blocking 2 days after exposure. And this effect is mediated, at least in part, by reduced corticothalamic input. Okay, so what does this mean? So I'm just going to leave you with a lot of questions. So there's some positive, some exciting, interesting stuff. So one is that does psilocybin enhance tuning to or stimulus outcome associations with stimuli that our brain usually ignores? Because clearly we are ignoring the unblocked, the siesta, the salad, because we're focusing on the oysters. But somehow psilocybin is forcing us to pay attention to the salad. Okay. So that's one option. The other critical thing is that for any associative connection to occur or to change, so you already made an association and you want to change it, it's really important that the outcome provokes the animal to a backward scanning of the memory store. So you're not going to change an association unless you go and deal with your priors. So does psilocybin actually change the structure of how the animal does this scanning? So I think this is a complex question. I feel like probably only large-scale unit recording could potentially answer this. So it'll probably take many, many years for us to answer this, but I think that's very much a critical question in this context. And then, so I think, but what's exciting is that moving forward, at least for us, these behavioral tools can very much help us understand how psilocybin can modify behavioral flexibility and how it can actually potentially relax priors. So we actually have behavioral tools that can quantify certain constructs, and then we can get to the mechanism. Okay, but there are also negative takes on this. And one is that the Kamin blocking is used extensively in humans, and because it's associated with, it's attributed to shifts in disruptive salience attribution and prediction error signaling. In humans, unreality beliefs, and also in individuals with acute schizophrenia, they have much weaker Kamin blocking. So this observation could also model psychosis or potentially adverse delusion-like beliefs and behaviors. So in terms of which it could be modeling, the jury's out, but something that is definitely on our mind. Okay, so with that, I am grateful for my incredible lab. And this is the current crew in Oregon. The people, again, that very much contributed to this are David Jacobs, Alejandro Torrado Pacheco, and Elena Bokachuk. I'm grateful for funders. None of this work is funded by NIH, but some private foundations. But I'm going to thank you, and then I'm happy to answer any questions. And I have other data, especially related to other conditioning things. If you're interested, I'm happy to share them. Thank you.